Welcome to your last video in your Chapter 12 video lecture series, where we're going to talk about Section 12.5 on communication between neurons. So we've already talked extensively about graded potentials, but I just wanted to make sure that we understand all aspects here. So when we have depolarizing graded potentials, we're often going to find it as a result of either sodium or calcium entering the cell. We'll talk more about sodium when it's entering the cell of neurons or skeletal muscle, but calcium plays a role in cardiac muscle cells. And the inside of the cell is going to become more positive. So let's take a look at some of these. Here we're going to see a small stimulus that is causing a small depolarization. Here's a larger stimulus, still depolarization, and here's a stimulus of a longer duration. So this is going to cause a longer lasting depolarization, but we see that it's not strong enough to reach threshold and cause an action potential. Now this one here is a larger stimulus that does reach threshold, and so this one is going to trigger a action potential in a postsynaptic neuron. Now for our hyperpolarizing graded potentials, these can be caused by potassium leaving the cell or chlorine entering the cell. And the inside of the cell is going to become more negative. So let's go back to our resting membrane potential, and we can see here that we have some stimuli that is resulting in hyperpolarization. And this is going to depend on the specific ion channels activated in the cell membrane and makes it more difficult for us to have a stimulus to allow us to reach threshold for an action potential to take place. Now, a postsynaptic potential is a graded potential in the dendrites of a neuron that is receiving synapses from other cells. So we can see that by these axon terminals here at these dendrites in the green. If we have a excitatory postsynaptic potential or an EPSP, this is depolarizing. So depolarization is happening within the postsynaptic potential, and you can see these in green on our graph. And again, at the green of these axon terminals synapsing onto the dendrites, and this causes the membrane potential to move toward the threshold. So let's say that threshold is at -55. And then we can also have inhibitory postsynaptic potentials or IPSPs. These are essentially hyperpolarizations that can take place in a postsynaptic potential and causes that membrane potential to move away from the threshold. So it's bringing it back down. And we can imagine that they are these axon terminals here at the axon hillock. So what happens is we have to kind of do summation here. You're adding all of these potentials together to see which is more stronger, our EPSPs or the IPSPs. Which do we have more of? So it looks like toward the end here, we have enough of those EPSPs to bring us up to threshold and allow an action potential to take place. So let's talk more about summation. All types of graded potentials are going to result in those small changes that we saw in that graph that are either depolarization or hyperpolarization in the voltage of our membrane. And these changes are going to lead to the neuron reaching threshold if all those changes add together or summates. So we have two types of summation that we're going to talk about. We have spatial summation, which is associated with the activity of multiple inputs to a neuron at different locations. So for instance, here are two different neurons. They're both sending EPSPs over to my second neuron here, or third neuron, I guess I should call it. And they're synapsing at different locations, but we are still summating. So here is the EPSP of this, let's call this neuron A. Here's the EPSP of neuron B. And together they summate, reaching threshold, so we can send that action potential through neuron C here. With temporal summation, we are going to have it as a relationship of multiple action potentials from a single cell all at the same time. So these stimuli are happening more frequently in order to send those signals. And so they are happening really quickly back to back so that this EPSP doesn't have a chance to go back down to its resting membrane potential. And instead, we get another EPSP right away and they summate. They add together and we reach threshold for that action potential to be moved through this second neuron. And now for a little bit more detail on synapses. There are actually two different types of synapses that we can talk about. We have chemical synapses, which is a chemical signal, namely through these neurotransmitters that are released from one cell and it affects another cell, or postsynaptic neuron in this case. An electrical synapse is when there is a direct connection between two cells so that the ions can pass directly from one cell to the next. So that would be something that would take place, let's say, in a cardiac muscle cell. And so some of the characteristics that are going to have to take place within a synapse is this presynaptic element, our neurotransmitter, a synaptic clef, which is a space in here, receptor proteins, our postsynaptic element, and something to eliminate neurotransmitters like an enzyme or reuptake. So I know this was all labeled in the previous slide, but this is in your guided notes, so make sure to label these. We are going to have an action potential move through this presynaptic neuron and reach the axon terminal. Now, when that happens, we are going to allow calcium-gated channels to open, and calcium will move and bind to these synaptic vesicles, which are going to contain neurotransmitters. And these neurotransmitters are then going to move toward the end of this axon terminal and exocytose, or go through exocytosis to release those neurotransmitters into the synaptic cleft, which is the gap between these two neurons, or this space between the two neurons. These neurotransmitters are then going to diffuse to the postsynaptic membrane and attach to these ligand-gated channels with the receptors for the specific neurotransmitter. So this is our chemical synapse. This is a chemical event. And the last thing we're going to discuss are some disorders of the nervous system. For instance, in Parkinson's disease is a chronic neurodegenerative movement disorder that affects voluntary and emotional movements. This is going to be linked to an increase in a protein known as alpha-synuclein that is toxic to our cells, and specifically in an area known as the substantia nigra in our midbrain of our brain. And you can see that highlighted over here. The midbrain is within our brainstem, and some of the symptoms that we're going to see are cognitive. So we'll lose some of those executive functions that allows us to plan or make decisions or control our emotions. Physically, we said this was a movement disorder, right? So they may have tremors that are especially noticeable within the hands. There will be stiffness within their body and slowed movement. So you'll often see people with Parkinson's, when they walk, they have kind of more of a shuffling movement. And then inside the brain, that substantia nigra has these special neurotransmitters known as dopamine, and we start to lose that dopamine production there. And then if we are looking inside of our neuron here, we have these Lewy bodies that are clusters of those alpha-synuclein proteins that start to accumulate there. Now for Alzheimer's, this is a neurological brain disorder, and it's the most common form of dementia that impairs mental functioning. And this is linked to another accumulation of proteins. Specifically, they are going to create beta amyloid plaques that are dense conglomerations of a protein that is not functioning correctly. Interestingly enough, when you go to sleep, your microglial cells, remember those nursing glial cells within our central nervous system, they help to get rid of beta amyloid. So the more sleep you get, the better, so that we can avoid these plaques from occurring later in life. So just a little side note. And those accumulations are going to be within the cerebral cortex. So symptoms of people with Alzheimer's are going to be cognitive, where they'll have memory loss and deterioration in thinking and planning functions. And physical changes could include slowness or rigidity or tremors as well. And areas that are affected are the cortex, but specifically this area of the hippocampus that are so important into memory and planning. So this area is going to shrink. And our ventricles are also going to enlarge. Those were those spaces that were filled with cerebrospinal fluid. And then this is showing us some of those beta amyloid plaques that develop. And those plaques are not going to allow these neurons to communicate with one another, which also is going to lead to impaired mental functioning. So the last disease we'll discuss is the Creutzfeldt-Jakob or Jacob disease, however you want to pronounce it. This is the human variant of the prion disease that is known as the mad cow disease in bovine. So a lot of people don't understand what a prion is. We are still learning more and more about prions. They are essentially, if you're going to compare it to anything that maybe you're familiar with, is that they are sort of like a virus, but smaller, and basically they're proteinaceous. They're not going to have like parts of a cell, like a nucleus or anything like that. They are going to cause this disease that is rapidly progressive, fatal, neurodegenerative, which is caused by that prion, and it could lead to things like dementia and death. And they also are going to have an accumulation of amyloid plaques.